Today, I'm going to be walking you through some of the top 10 histology question and answer series as the first episode we are going to be releasing. And based on this top 10, we would like that you follow it up and comment in the section of the comment area to tell us exactly how many you had correct out of the 10 we are going to be sharing. In fact, we are going to give some various options of correct answer for you to choose among the list. Then tell us in the comment section which among you have correct. If you have, for example, 6 out of 10, just comment 6 out of 10. Be honest and let us know to check your skills in terms of these histology questions. So beginning with the first, to those of you who want to have an update on the next upcoming session of this episode to see the next possible list of questions and answers we're going to be releasing very soon, then you want to make sure that you go ahead to subscribe and also enable the notification bell icon so that anytime we release the new episode or series, you'll be the very first to watch and see what new we have as an update. So without wasting time, let's look at the first possible question here. Since we are dealing with histology, let's first of all begin with its definition. So based on it, histology is basically defined as the study of, we have four different options. A, we have structure of organs visible to the naked eye. B, chemical reaction in living cells. C, microscopic structure of tissues. And D, genetic material and inheritance. Which of these options is correct? In fact, if you look at this, histology is actually talking about the C option, which is the microscopic structure of tissues. Microscopic structure of tissues is all about histology. So the correct option here is option C. In fact, let's now move to the next question. The next question, as the second option, we are looking at the basic structure and functional unit of all tissue is known as A, tissue, B, cell, D, organ, and C, organ, and D, system. Which is correct here? Choose the correct option. So for us here, the correct option is B, which is cell. In fact, the basic structure and functional unit of all tissue is known as a cell. So the correct option is B, which is cell. Let's look at the next question here. The next question, as question number 3, we are looking at which of the following is not a basic type of tissue. Which is not a basic type of tissue? We have option A, epithelia, option B, connective, C, nervous, and D, vascular. In fact, the first 3 are all tissues, but the last one, D, which is vascular, is not. So the correct option here is D, which is not. Let's look at the next question. The next question, as option 4, will be which microscope is commonly used in routine histology laboratories? A, the electron microscope. B, the fluorescence microscope. C, light microscope. And D, confocal microscope, which is correct here. In fact, when we talked about microscope being used is in most histology laboratories, then of course we are talking about option C, which is the light microscope. Because the light microscope is the standard tool in histology because it allows viewing of stained tissues sections, and also because it's practical and affordable and also sufficient for most diagnostic purposes. That is why it is the light microscope is the correct option. Please remember the option that I've given to you. Let's move at the next question, question 5. The fifth question states the usual thickness of histological sections that is prepared for light microscope is which diameter? Is approximately which diameter? A, we have 0.1 micrometer in diameter. B, 1 to 2 micrometer in the diameter. C, 5 to 7. D, 20 to 30. In fact, you need to know the correct option here. First, make a choice. But note that the correct option here is C, which is between 5 to 7 micrometer because it allowed light to pass through while maintaining tissue integrity. If it is very thick, that is between 20 to 30, it will be very thick in a way that it cannot allow the passage of light through it for us to conduct the lab light microscope to see all the things we need to see. So the thinner the section, that is, thinner sections are used for electron microscopy. In fact, option A, which is 0.1, is not very correct because it is very, very thin. And B also thin, but the line range is 5 to 7 micrometer in its diameter. So the correct option here is option C. Let's move to the next question, which is question 6. Question 6 states, which staining technique is mostly commonly used in routine laboratory or routine histology? Let's use the word histology. Which staining technique is mostly commonly used in routine histology? We have A, the pass stain, B, hematoxylin and eosin, that is H and E. Then we have C, which is silver stain, and D, which is giemsa stain. In fact, choose the correct one, but note that the correct one here is B, which is H and E. You should note that H and E stand for hematoxylin and eosin. This is the exact technique of staining which is used, H and E staining. So it is correct, which is because the hematoxylin stains the nucleus or nuclei blue and purple, while eosin stains pink. So these are the stains which are mostly used, the H and E stain. While the H is blue and purple, the E is pink. Let's look at question 7. Question 7 option. We have hematoxylin, which we have just seen before, is considered A, acidic dye, B, neutral dye, C, basic dye, and D, lipid dye. In fact, choose the correct one, but note that it is the C option, which is basic dye. In fact, that hematoxylin is basic. It is basic dye, and so it binds to acidic structure like DNA and RNA, because if we are conducting any test using the electron microscope, using the light microscope for it to stain to see what we want to see, then of course since RNA and DNA are mostly structures that tend to be acidic, then the stain, which is A and H and E, that is hematoxylin or eosin, must be what is called termed as, it must be in the term of a basic, because basic and acidic could work together. If the stain is basic, then of course the strand itself, which is DNA or RNA, have to be acidic. So this is the sense knowledge of it. So we should choose option C, which of course in this case it is basic dye. Let's move to the next option. The next option is option 8, that is question 8. We're looking at eosin primary stains which structure? They are given A, nuclear. They are given B, acidic component. They are given C, cytoplasm and extracellular protein, and they are given D as lipid. In fact, the correct option here, even as you have chosen, but note that the correct option is C, which is the cytoplasm and extracellular protein. In fact, eosin is an acidic dye, and because it is an acidic dye, it needs to bind, therefore, to, that is, basic component such as cytoplasmic protein and also collagen fibers. This produces the characteristic pink color which we are looking at, because of course the eosin, remember, is going to stain pink once it meets that is acidic strands or acidic medium or any acidic nature like the cytoplasmic protein or maybe collagen fiber. Any acidic substance is going to stain the pink. So that is why option C is correct. Let's move to the next question, question 9. Question 9 is going to state, fixation of tissues is important because A, it enhances staining, B, it prevents autolysis and also decomposition, C, makes tissue to be thinner, and D, it removes water. Which is correct among this list? In fact, you need to note that the correct option, despite what you have chosen, the correct option here is option B, which is, of course, prevent autolysis and decomposition. That is the importance of fixation. This is because fixation helps to preserve tissues by how? That is by killing cells and also preventing enzyme or enzymatic digestion, and also maintaining structure close to life. In fact, that is the importance of how the B part of it, or how fixation actually help in preventing the cell from itself being killed. That is, fixation preserve tissue by killing the cells. That is how it preserve it. Second way, making sure that it prevents enzymatic digestion. And the third way, maintaining structures that are close to life. And therefore, formalin is the most common fixative. Now, let's move to question number 10. Question number 10, we're looking the most commonly used fixative in histology is 1. Alcohol. B. We looked at glutaraldehyde. We looked at formalin, and we looked at acetone. Remember, I've just told you in the last question that it is mostly formalin that is being used as a fixative in most labs to prevent decomposition of cells, to prevent killing of the cell itself and those cells that are close to life, to maintain it in that way, to prevent it from going away. So, of course, if we are talking about which particular fixes is being used in lab, then note that it is actually formalin. You cannot use acetone, you cannot use alcohol, you cannot use glutaraldehyde, but the only option here is the formalin, which is used as fixative to preserve these particular cells or tissues from decomposing, or from making sure that they undergo self-digestion. In fact, we have other most important top questions we are going to be sharing very soon. In fact, if you want to make sure that you see the next upload, you want to make sure to subscribe and enable the notification bell icon so that when we publish the most valuable upcoming top 10 questions and answers for this session, which is going to be the next episode, you are going to be the very first to receive the notification to watch, to see the update we have for you. You also want to make sure to leave a like to tell us that you like what we do, and see you again in the next upload, and bye-bye for now.